Hello, my name is Andrea O'Hara, and I am one of our technical specialists on the GENEWIZ NGS team. And today I'll be presenting to you Advancing AAV, Novel Synthesis and Sequencing Solutions for Gene Therapy Research. At GENEWIZ, we have a number of different proprietary protocols for both sequencing and synthesis of AAV. This includes solutions for the upstream discovery and identification, preclinical development, and downstream clinical development and use. And within these, we have a number of different solutions at our disposal, including solutions around next generation sequencing, Sanger-based services, and synthetic DNA solutions. I'm going to take you throughout the AAV lifecycle and introduce some of the different solutions we have to offer that encompass this entire spectrum. Adeno-associated virus, or AAV, is an ideal delivery vehicle for gene therapy. The AAV genome consists of a left and right ITR, as well as the rep and cap proteins. However, this molecule can be utilized to insert our own targeted recombinant gene or genes of interest. The packaging size is limited to approximately 5 kb, but one thing to keep in mind is that these left and right ITRs are critical for virus replication and encapsulation. In fact, maintaining the ITR throughout production is critical for the entire process for AAV quality and efficiency. There's been a number of publications that have covered this in great detail, but the ITRs are really important for AAV productivity, encapsulation, residue of non-recombinant AAV encapsulated DNA, the affinity of the rep protein, and transduction efficiency, just to name a few. A key highlight, if left undetected, would reduce reproducibility of experiments both within and across laboratories and potentially cause significant delay of vector translation into clinical utility. So what are some of the main challenges when working with the ITRs? The first is actually sequencing the ITR. The ITR region itself, which is shown here on the upper right-hand side, consists of this T-shaped hairpin loops, which makes it very difficult to sequence. And we need to know what is the actual sequence of the ITR on the plasmid. The second component is that the ITRs are really prone to mutation during plasmid preparation. So how can we get a good batch of AAV plasmid without ITR mutations? Enter GENEWIZ. We have a number of different AAV solutions to ensure a good start for high quality AAV manufacturing. This includes our proprietary Sanger sequencing method that can read through the ITRs, ITR correction and AAV plasmid synthesis, and finally AAV plasmid prep. We also offer some next generation sequencing solutions for the AAV ITR sequencing. I'd like to take you through these in a little bit more detail. So the GENEWIZ ITR sequencing is able to actually read through a full-length ITR. The existing Sanger sequencing method quickly drops off throughout that 125 base pair hairpin loop. However, with our proprietary sequencing method, we can read cleanly throughout the entire 125 base pair region. On the left-hand side is showing existing Sanger sequencing methods, and you can see how that sequence really just drops off. With our proprietary method, we're able to read through the entire 125 base pair region of that hairpin loop. This is an ideal approach for confirming your plasmid sequence and making sure that you have great ITR fidelity. We do also offer this at the GLP grade. So the GLP grade AAV ITR sequencing confirmation is FDA compliant and includes PCR amplification and Sanger sequencing of the ITR regions using our proprietary protocol. Additionally, GLP grade sequencing confirmation includes QA oversight, a study report, signed GLP compliance statements, and archival study analysis of up to 5 years at a minimum. Looking for a next generation sequencing option, we also recommend our PacBio approach, which is a great alternative for high throughput screening and contamination testing of your ITR sequences. This is going to actually be sequencing of the entire plasmid. So this will include the ITR region as well as the rest of the plasmid and the backbone. This is a contiguous single long read molecule, so we'll get the whole plasmid with high accuracy. Again, single molecule resolution means that we can look at a mixed frequency population or a clonal population to confirm the clonality of that population. And we can multiplex multiple plasmid preps for high throughput testing. This is a really ideal approach to confirm that you have high fidelity of your plasmid stocks, that there is not any low-level contaminants, and if there are, to determine what they are. Now, once we have our ITR sequence known, we have a full plasmid preparation workflow to confirm, maintain, and correct the ITR as needed. So again, starting out with that AAV ITR Sanger sequencing approach, if we have a mixed population, we can do screening and selection to isolate the clones that have the intact ITR and stop propagating, remove those that have any sort of truncated or mutated ITRs, and carry those through to the plasmid preparation. If we are looking to actually synthesize a plasmid or make plasmid edits, we can clone intact ITR regions to it. Or if we have a plasmid that's been put together, but we notice that there are some inconsistencies within the ITR, we do offer ITR correction to fix these regions before cloning or scale up. Once we have a great plasmid that is full, intact, and ready to go, we have a number of solutions to take us moving forward. So again, we have sequenced our ITR and corrected it as needed. Now we can move on to our actual plasmid preparation. And again, you'll remember there's two main steps here that are really critical for this ITR. The first is confirming the sequence, but the second is maintaining that correct ITR sequence throughout plasmid preparation. And so to take you through a little bit more detail on this, I'd like to walk you through a case study. So at GENEWIZ, we have several proprietary cell lines that can best maintain ITR integrity throughout the plasmid prep process. In this case study, we took 8 different AAV plasmids and transformed them across 5 different cell lines. 2 of them are our GENEWIZ cell lines, which have been optimized for maintaining the ITR across the entire process, as well as 3 other companies' proprietary cell lines. For each of these, we picked 8 clones from each plate and then did the ITR sequencing with our Sanger method to confirm the ITR integrity. What we found is that the GENEWIZ proprietary AAV strains work best for AAV plasmids, especially those with easy to mutate ITRs. Among the 8 AAV plasmids that were tested, 3 were particularly challenging in terms of ITR integrity for plasmid preparation. Both GENEWIZ AAV strains can obtain clones with intact ITRs for all 3 plasmids, whereas the other commercial strains fail to generate clones with intact ITRs for at least 2 of the plasmids. So you can see here on the left-hand side, we have the overall ITR integrity, and on the right, the number of clones that actually have that intact ITR. What we can see here is that altogether, GENEWIZ has a greater than 98% success rate after just the first attempt for plasmid prep using these proprietary cell lines. Now, once we've generated our full intact plasmids, we're ready to move ahead with packaging of the cell line and generating final AAV viral particles. We're going to take all of these pieces together and generate the AAV particles, but once we have these AAV virus particles, we need to confirm that we have everything in there exactly as specified, no more, no less. That means that we want to confirm that we have the correct recombinant AAV viral genome packaged within the viral particles, and that we don't have any impurities or contaminants also packaged within these AAV virus particles. This is a really critical step for the entire pipeline because impurities packaged within those recombinant AAV vectors can seriously affect clinical safety. Impurities can include things like defective particles, residual proteins from the helper viruses, or contaminant DNA from multiple sources, including the plasmid backbone itself, the residual cell DNA from the growth, or the helper viruses. I'm highlighting here a few key publications on the right-hand side. But the main take-home message here is that it's imperative to confirm packaged material, since the introduction of incorrect sequences can trigger unwanted immune responses, and measuring the level of DNA contamination prior to clinical use can help reduce the risk of adverse side effects. To get to this, we have 2 main applications for next-generation sequencing post-packaging. The first is with Illumina sequencing, which is a short read technology, and the second is PacBio, which is a long read technology. I'd like to take you through the details of both of these, as there are pros and cons to both approaches. So as I mentioned, Illumina is a short read sequencing technology. With this method, we will go for a very high depth of coverage, but again, we're going to be using short reads that will be bioinformatically mapped back to our reference genomes. This is ideal for checking contamination and quantitation of those contaminants within that population. Again, we have two main focuses: confirm what the recombinant AAV virus is and see if there's anything else in there. Because we're doing this very deep sequencing, we can get resolution at a very low frequency of potential contaminants. And this again can include the backbone, helper plasmids, as well as any residual cell DNA. We're also going to be able to get a really nice deep view of our recombinant AAV genome. And so we'll be able to quantitate any mutations, small insertion deletions, and edits that might be occurring within our AAV genome. When we think Illumina, we think quantitation. Again, deep sequencing, so we're going to get a great idea of the frequency of these changes that we might have or contaminants present. Conversely, we also have PacBio long read sequencing. This is going to be single molecule resolution. That means that for every piece of DNA we have, we're going to sequence the full thing from end to end. This could be short DNAs or long DNAs, depending upon what's inside of those viral particles. We're going to be doing a mixed population analysis of this pool of viral stock. Our depth of coverage here, though, is going to be more moderate. So this approach is ideal for confirmation of our sequence fidelity. Again, because we're looking at the entire molecule end to end, we're going to be able to identify mutation events, recombination events, fusion events. We're also going to be able to look at truncation events within our ITRs or even earlier. And we're going to be able to read cleanly all the way through that 5' and 3' ITR. When we look at our full-length species of our recombinant AAV, we will be able to identify the abundance of variants, but again, we're not sequencing as deeply, so we won't have that ultra-deep resolution for very, very low-level variants. The other thing we'll be able to do is resolve the size of our contaminants. So because we're looking at that single molecule resolution and going from full end to end, if we look at any contaminants that we may have present, we'll be able to see if they are all the same size as our recombinant AAV, or do we have smaller things that are contaminating our overall viral particle pool. One of the caveats with PacBio sequencing is that it does have a size bias, meaning that it's more, it will proactively or selectively sequence smaller species. That means that PacBio is not ideal for determining frequency of contamination or for determining the frequency of size of particles, but within a particular size range, we can determine frequency of changes within it. So whereas Illumina, we think frequency, PacBio, we think fidelity. So what is my overall molecule looking like? And if I do have any contaminants, what are the size of those contaminants? So an overview of these two methods. PacBio, we're going to get full-length AAV fidelity. Again, that entire genome, end to end, we'll be able to identify the frequency of the changes within the full-length genome and the size of our contaminants. Illumina sequencing is short read sequencing. It's going to be very, very, very deep. So we will get a really excellent view of the frequency of the changes, but because it's not that full molecule, we won't know if we have multiple changes happening in the same species or if these are independent species changes. We're going to get that frequency, but we're not going to get the full molecule resolution. And we'll have an excellent view of the frequency of the contaminations to a very low level. Ultimately, the best approach here is going to be using both technologies. Because of their strengths and weaknesses together, we're going to get the most comprehensive view of what our full-length AAV looks like, any high and low-level frequency changes, the amount of contaminants we have, and the size of the contaminants. And when we put this together, we'll have a comprehensive view of everything that's happening within that final viral particle pool. Now at GENEWIZ, we offer full end-to-end AAV NGS workflows. You can select for either PacBio, Illumina, or both. This workflow includes extraction from viral particles, second-strand synthesis or conversion from the single-stranded DNA to a double-stranded DNA, library preparation, sequencing, and bioinformatics analysis. Our bioinformatics analysis includes standardized reports. This will have detailed variant reporting. That's where we'll find out about the frequency of our changes, if any, within the recombinant AAV genome. We will also have advanced mapping statistics. This will tell us about host contamination, helper plasmid contamination, and AAV plasmid backbone contamination. And we'll be able to detect truncation hotspots, including the 3' and 5' ITR sequences. And so I'm showing you just a few examples here. And again, this is all included within that comprehensive workflow. We also do offer GLP grade viral gDNA or cDNA Sanger sequencing confirmation. This is compliant with the FDA, and this includes a few different options. So this would be PCR and Sanger sequencing verification, including genome integration and cell bank confirmation. The starting materials accepted include viral particles, viral vector, master virus stock, genomic DNA, mRNA, or cell pellets. And again, with the GLP grade Sanger sequencing confirmation, this entire project will include QA oversight, a study report, signed GLP compliance statements, and archival study analysis of 5 years at a minimum. Now, once we've completed our entire AAV packaging process, the next step are going to be our post-infection solutions. So again, thinking back to the entire end-to-end AAV workflow, once we've completed our initial discovery phases, we have our preclinical and post-clinical phases, or preclinical and clinical phases, if you will. We can use next-generation sequencing for a number of the post-packaging solutions during these preclinical and clinical phases. And I have this broken up into 3 main components. The first is going to be transcript or gene expression profiling, both during and after infection. The second is going to be about immune response during and after infection. And finally, characterization of the host genome, which is available at research use only in CLIA. The top 2 phases, I'm going to take you through all of these in more detail, but the first 2 are at RUO only. So the first one, the transcript or gene expression profiling during and after infection. We have a number of different sequencing solutions that can be utilized for gene expression or transcript expression profiling, both during infection and after infection. Bulk RNA-seq or standard RNA-seq can be used for things like treatment versus control or dosage response, and this is typically going to be Illumina-based RNA sequencing. We also offer single-cell RNA sequencing. This is really ideal for doing a much deeper dive, again, treated versus control or dosage response, but looking for single-cell resolution or cell type resolution of changes. Especially critical if we think we are going to be targeting specific cell types with our recombinant AAV vector, so that we can see where those changes are occurring, at what frequency, and is it having an impact on multiple cell types or differing impacts on cell types. We also offer PacBio IsoSeq, which is isoform detection and confirmation. Really ideal if our recombinant AAV is going to be providing a different transcript versus something that's going to be already present in the cell. The idea here being, instead of looking for something that is in addition to, or looking for a species that's in addition to another one that's already present versus say an example where we are introducing something that is no longer present with our novel gene therapy. Finally, we have digital spatial profiling, or DSP. This is on the nanostring genomics. And this is ideal for in situ identification of expression. With this, we would actually take a tissue sample and look for the expression within the tissue sample. Again, really ideal if we're thinking of targeting specific cells or cell types, and we want to see that within the 3D context of the tissue. Now we can use these similar approaches to actually look at the immune response and immune profiling both during and after infection. And this is really thinking about now looking at the immune components in particular. So all of these have options that focus more specifically on the immunoprofiling versus the standard expression profiling. And for each of these solutions, they are both a standalone or an add-on, meaning you can look at just immunosequencing alone, or you can combine immunosequencing with transcript or gene expression sequencing. Again, all of these services are at the research use only level, but we also offer some solutions that are going to be at the RUO and CLIA levels. And so here, this is where we're going to think about characterization of the host genome. This can be with whole genome sequencing at standard or low-pass whole genome sequencing. Standard whole genome sequencing is going to give us a high level of coverage. Low-pass whole genome sequencing is ideal for SNP level detection. This is also really ideal if we want to catalog our cohort for the downstream clinical trial. So if we're looking for, say, variability or differences in our full-on solution, or we want to get some SNP level genome profiling to identify additional markers. This also includes targeted sequencing, whole exome sequencing, or even targeted panels. All of these offerings are available at research use only or at the CLIA stage. And so to recap, we have a number of different proprietary protocols for both sequencing and synthesis of AAV molecules. From our Sanger-based services, this included AAV ITR sequencing at both our research use only and GLP compliant levels. Our synthetic DNA solutions included both DNA synthesis, gene synthesis, plasmid synthesis, as well as plasmid prep. Finally, we offered a number of different next generation sequencing solutions. And this included whole genome sequencing of the recombinant AAV vector itself, as well as whole genome sequencing solutions for the clinical trial and preclinical stages. Additionally, we offer a number of downstream approaches for the post-infection solutions within the next generation sequencing stages. At GENEWIZ, we're here to offer you complete and comprehensive full end-to-end sequences or full end-to-end solutions. And so with that, I would like to actually go ahead and take any questions that we may have. Okay, and I see we have a number of questions, so I'm going to go ahead and just read these off. The first question is for recombinant AAV sequencing, how much single-stranded DNA do I need, or how many particles do I need? This is a great question. So for Illumina sequencing, we typically want about 500 nanograms of DNA at a minimum, and that typically corresponds to an area about 1e to the 11th to 1e to the 12th viral particles. For PacBio sequencing, this is a much hungrier technology, so we typically need more DNA. And so for this approach, I recommend 1 to 2 micrograms of DNA per sample. And so that corresponds to closer to a neighborhood of 1e to the 12th to 1e to the 13th particles. If you're looking to do both technologies for your viral vectors, I do recommend shooting for that 1e to the 13th viral particles. Again, we offer full, that package is a full package, so you can either provide the single-stranded DNA after extraction, or you can just provide the particles and we can do the extraction as a part of that comprehensive workflow. Okay, the next question is how much does it cost? So it really depends on which approach you're working for here. So obviously we have a number of next generation sequencing solutions as well as Sanger sequencing solutions. I do recommend, if you're interested in learning more about cost, about the different approaches, if you're thinking of using multiple different approaches, that the best place to go is to our website, GENEWIZ.com. We do have an AAV solutions webpage. Where you can actually click through and select each of these different technology options, including both the Sanger and next generation sequencing solutions. And you can read more about it, but also you can request pricing and quote information on our website. Okay. And with that, I think we'll actually wrap up here. So if you do have any questions that I didn't answer or you would like to discuss further, I would invite you to visit us at our booth. I will be there throughout this conference to answer any questions that you may have. So please feel free to come over to the booth and reach out. You can also get additional information at our booth regarding any of the solutions you've seen here, as well as some of the other solutions that we do offer at GENEWIZ. And with that, I will go ahead and say goodbye. Have a great day, everybody.